Egypt, November 2015. While vacationing with his wife, Tom Patterson, a professor of psychiatry at UC San Diego School of Medicine, contracted a life-threatening infection with a multi-drug resistant strain of Acetobacter baumannii. The following year, in 2016, his wife, Stephanie Strathty, enlisted the help of an international team of physicians and researchers to save her husband's life using bacteriophage therapy. Dr. Patterson made a quick recovery and appears to be the first person in the United States to be successfully cured from a systemic multi-drug resistant bacterial infection utilizing cocktails of intravenous bacteriophages. Today, we're going to talk about antibiotic resistance, an extremely pertinent problem that is going to continue to affect our healthcare industry for decades to come. Data from the Centers for Disease Control has concluded that over 2 million people are sickened every single year in the United States by drug-resistant strains of bacteria. And of those 2 million, approximately 23,000 will succumb to their illnesses and pass away. Now, this problem of bacteria evolving resistance to our medicines is definitely exacerbated by the overprescription of antibiotics. A report in the British Medical Journal found that out of 19 million privately insured outpatients in the United States, approximately 1 in 7 was being prescribed pointless antibiotics that they did not need and were not necessary for curing whatever ailments they were suffering. But whether we speed up the problem by overprescribing antibiotics, or we try to slow down the problem by reeling in our overprescription, at the end of the day, bacteria are still evolving resistance to the 26 different medications that we currently rely on. And as more and more of them become useless, the extra $2.2 billion that we're losing in healthcare costs is only going to increase. So the demand for new antimicrobials that take an innovative and novel approach at taking down these new superbugs of bacteria is at an all-time high. And one of these methods that is gaining some traction and popularity is the use of phage therapy, which actually has been around for a pretty long time. Being in use since the early 1900s and being more popular in other regions of the world like Russia, the word phage is actually short for bacteriophage, which just means a virus that infects specifically bacterial cells. Phage therapy is the practice of using these bacteria-specific viruses to treat bacterial infections inside of patients. But if the anti-vaccination movement has taught us anything about the general public, it is that they can be very distrusting of using viruses in any capacity as therapeutic treatment. So if we're going to push for a movement of phage therapy as a novel approach to dealing with the antibiotic resistance crisis, it is going to be critical to start an educational campaign that can inform the public on how phage therapy works and why it is a safe and potentially effective practice. Viruses are obligate intracellular parasites. It's basically a piece of DNA or RNA that is encapsulated inside a protein coat. They need a host cell to thrive and reproduce. Every cell in our body has a series of surface proteins that are found on the surface of a cell membrane. And the protein coat of a virus has a series of receptors that are specified to their target host surface protein. In other words, it's like a lock and key mechanism. Each virus has a series of proteins on its surface that are designed specifically to match up with its host. So what does that mean? Well, phages, or viruses that infect bacteria, have specific protein coats that are designed to bind to bacterial cells and inject them with their viral DNA or RNA. Those viruses are very, very specific to their specific strain of bacteria, and they essentially ignore everything else, us included. So you are actually surrounded by billions of viruses every second of every day. Most of them just aren't targeted to you as a host, so they don't do anything to you. And so our scientists have said, hey, if we can take these specific viruses that infect X strain of bacteria and use it on a patient, these viruses should, in theory, infect and kill that specific strain of bacteria leaving the host relatively unharmed and eradicated of their bacterial infection. And in Dr. Patterson's case, it was actually proven to be quite useful. Now, the idea of implementing phage therapy does come with another hitch in the works. The US Food and Drug Administration. Current FDA regulations are not the friendliest for encouraging this technology to flourish in our society, which is why it's widely been ignored and not used in the West. Traditional Food and Drug Administration regulatory protocols for antibiotics requires that all components of any new drug go through individual clinical trials, and the recombination of these multi-cocktail drugs can't occur without reapproval. And FDA approval for a drug is a lengthy and costly process. Typically, our chemical antibiotics work in a kind of broad spectrum approach by using a specifically designed molecule that binds to and inhibits certain enzymatic activities that are going on inside of the bacterial cell, largely that are designed with making the peptidoglycan strands of a bacterial cell wall, which ultimately causes these bacteria cells to burst due to osmotic pressure. It is a topic that I actually talked about before in my video on the endotoxin detecting properties of horseshoe crab blood. Phage therapy is very, very, very specific. You have one specific virus that goes after one specific strain of bacteria. In fact, phage therapy is so specific that generally it's been considered a personalized medicine. Taking a sample swab from a specific patient and running bioinformatics on the specific strain of bacteria they have, then going to a phage library and picking out the phage that is best targeted to take on that specific bacteria. In order to use phage therapy ever as a more broad spectrum approach, you would need a pretty significant phage cocktail where you are utilizing multiple viruses to try and cover multiple strains of the pathogenic bacteria. And by current FDA regulations, if you're going to make a cocktail of all of these phages, every single phage individually on its own would need to undergo clinical trials for safety screenings. And quite frankly, that just hasn't been worth the time or the cost to go through as a procedure. It's simply not practical. And yet, there is a solution to this problem that is standing right in front of our faces, and it comes from your flu shots. The FDA could opt to manage its regulation over phage cocktails in a methodology that is analogous to the way that it regulates vaccinations. The influenza vaccine that can prepare seasonal flu shots compiles data from the Centers for Disease Control and determines what the 3 or 4 or 5 most commonly circulating influenza viruses are in a given year. They then use that information to construct their seasonal flu shot, which contains non-pathogenic forms of those viruses. But logistically speaking, it would be virtually impossible for these organizations to conduct individual clinical trials for every single strain of influenza virus that might circulate and affect a population in a given year. There's just not time to do that and get the flu vaccine out for people on time. So what the FDA has done instead is they approved the process by which the vaccines are developed. And phage cocktails are actually prime for being approved in this way. By approving the methodologies, the FDA could set standardized protocols for the safety of phage cocktail preparation. They could set standards for only utilizing lytic phages and ignoring any phage that would be lysogenic, that being one that would actually alter the DNA of the host. They could standardize the process of purifying these phages, things like removing the endotoxins that could cause fever-like symptoms, and the selection of appropriate phages for each patient's individualized condition. The FDA is the key here to this puzzle. Their approval of a more streamlined system for phage cocktail approval is the thing that will pivot the direction and allow more and more established clinics in America to begin running with these procedures. Furthermore, the feasibility of this is being showcased by an emerging biotechnology company that is setting the standard of innovation. Locus Biosciences is a corporation that is combining the basics of phage therapy with CRISPR-Cas technology to develop new and novel precision-based antibacterials. Now, I typically talk about CRISPR and its association with Cas9, CRISPR-associated protein 9, an endonuclease protein that is guided by CRISPR RNA to specific sequences of DNA, and then it creates a cut, just like a pair of molecular scissors. Scientists were able to manipulate the Cas9 protein by creating their own guide RNA sequences. We learned that in laboratories, you can essentially stitch together any letters you want, and you can tell Cas9 where to go. The scientists at Locus Biosciences, however, use a different Cas protein. They use Cas3, which is also an endonuclease. It is also guided to specific sequences of DNA by a guiding RNA strand that the scientists can construct in a laboratory to tell it where to go. Where Cas3 differs is in the way that it works. When Cas3 binds to its target sequence of DNA, rather than creating one single cut that can then be repaired by endogenous DNA repair pathways, Cas3 acts like a DNA shredder. It just cruises along the DNA strand, shredding it to bits and pieces that are beyond repair. It's great for completely knocking out sequences of DNA. And the scientists at Locus Biosciences are using this Cas3 protein and guiding RNA strands specifically to fight off the development of antibiotic resistance. Essentially, they're using the Cas3 system to supercharge their phages and make sure that they can continuously, efficiently knock out these bacteria. And this company is getting involved in some serious business. In 2018, the company acquired Epi Biomes' high-throughput discovery platform, which utilized high-throughput screening to find novel therapeutic phages. High-throughput screening is basically automated screening technology services. Rather than a scientist standing in a laboratory and pipetting the thing into the thing, when you automate that process using robotics, you can do these experiments on a large scale with a greater efficacy for replicability. And so now, by utilizing one of the world's strongest automated phage discovery platform, Locus Biosciences has an incredible basis for developing broad-spectrum phage cocktails. And they're capturing the attention of investors too. Locus entered a collaboration and license agreement with Johnson & Johnson for the development of their Cas3 mediated phage therapy. And in this year, 2019, it's been announced by Locus Biosciences Chief Executive Officer Paul Garofolo that they are preparing to enter clinical trials for testing a new CRISPR phage therapy on E. coli infections of the urinary tract. By taking a dual mechanistic approach at eliminating bacteria in patients, this CRISPR phage therapy could really revolutionize the way that we utilize antibiotics, and it could be one of these solutions that we need for our antibiotic resistance crisis. And that's pretty cool. So, as always, thank you for watching and stay inquisitive. Bye.